So welcome everyone to this Physics of Life seminar, which will be given by Professor Ned Wingreen from Princeton University. So before we get started, I just want to give a couple of, I want to go through a couple of housekeeping rules. Please mute your microphone so that we don't have any unwanted feedback. But do submit questions on the chat box, and we will try to ask them during the talk. And if there's time and Ned sticks around a little bit, we can also have more discussion afterwards. And note also again that the talk is recorded. So I don't want to waste any more time. So it's a great pleasure to have Ned Wingreen here. He's a Howard A. Pryor Professor in Life Sciences at Princeton University in the Department of Molecular Biology and the Louis Sigler Institute for Integrative Genomics. He did his undergraduate work at Caltech, and then he went on to do his PhD in physics at Cornell University, and he spent also some time at KITP in Santa Barbara and MIT, if I'm informed correctly. And then in particular, during his tenure at NEC Research Institute, I wanted to say laboratory, I think they changed the name a few times. He's then switched from semiconductors and conventional physics to biological problems. And there's actually a kind of a nice little online talk he gave with DBio, the division of biological physics from APS, about his career. So it's a little bit hard to sum up all the things Ned has done over his careers, but he certainly contributed a lot of fundamental understanding to intercellular networks in bacteria and more recently in microbial communities. And maybe most importantly, he's a fantastic mentor and teacher. And yeah, he really pushed the field forward over the years. Maybe one thing to note also is that he did fantastic teaching, for which he also won some awards. In particular, that methods in logic and quantitative biology. And I think I was sitting in some of these classes when he started to develop the course around, I don't know, 2005 or something like that. So that was really fantastic. Yeah, so I'm really happy that he's here. And without further ado, yeah, please, Ned. I'm looking forward to your talk.
Thanks, Robert, and thanks for the terrific introduction. I miss the days when you and I were just learning about biological physics and getting into the field. Those were fun times. But thanks everyone for joining today. I'm excited to tell you about this field of protein phase transitions and some of the work we've been doing, specifically having to do with a topic in magic numbers. Let me start though by telling you, what the plan is for today, and I hope that people will feel free to ask questions in chat. We'll have a conversation afterwards. I will pause in the middle also to see if there are any questions. But I'm going to start out with what is necessarily a brief introduction to this, fascinating topic of phase separation and phase separated condensates inside of cells, and also introduce some of the work that's been done in vitro, outside of cells in glass to understand the physical properties of these condensates. Then I'm going to focus on a particular class of these condensates where there are two components, both of which are necessary for phase separation and that are multivalent, that have many binding sites on each component, because this gives rise to some very interesting statistical physics. I'll start out by talking about an in vitro system, the Sim Sumo system, which is engineered polymers, but I definitely want to leave time to talk about this amazing living system, something called the algal Pyrenoid. Now, I had vaguely heard of algae before, but I'd never heard of the Pyrenoid. It turns out we should all know and love the Pyrenoid because it's responsible for fixing about a third of all of the CO2 that comes out of the atmosphere every year. So we would all be in trouble if there weren't Pyrenoids around. And it's an example of one of these phase-separated condensates. And at the end, I'll try to draw some general conclusions from what we've learned so far. So going forward, I want to start with this rather busy slide that I've borrowed from my colleague Cliff Brangwyn, who's really one of the heroes of this field of intracellular phase separation, not only one of the discoverers, but ongoing one of the leaders. I have the wonderful experience of interacting with him and his group at Princeton. And there's a number of things shown here, and I want to make a couple of different points about what's going on inside of cells. So here, we have our kind of schematic of a cell. It doesn't look really that crowded. The real cell is, of course, tremendously crowded. but so that you can see it, there are a few things that we know about already, and we've known about for many years, which is that the eukaryotic cell, our cells, are divided up by membrane-bound organelles. So there's the nucleus, where our DNA is stored. There are mitochondria, sort of the energy factories of the cell, lysosomes, and various other bodies that are separated off from the rest of the cell by membranes. But what's become clear over the past decade or so is that this is only the tip of the iceberg of the intracellular organization inside of our cells. And what else is happening is lots and lots of phase-separated bodies that are not bound by membranes. They're not separated from the rest of the cell by membranes, but rather by physical interactions that hold proteins and RNAs together, much in the same way that oil and water separate, not because there's a special membrane that goes around the oil, not because of special motors that pull the oil molecules together, but simply because of the physical chemistry of the oil molecules preferring to be near oil molecules and the water molecules preferring to be near water molecules. And it's this same kind of physical chemical interactions that have given rise to all of these different phase-separated bodies that you can see on this plot. And I just want to emphasize that this really spans not only the space, that they're in the nucleus, they're in the cytoplasm, but also biological function. So increasingly it's become clear that phase separation is playing a role in fundamental processes like the transcription of genes, also in sequestering materials, keeping them safe for when they're going to be needed, but also in things like metabolism. So here's an example, beautiful example from Steve Benkovich's group, something called the pyrenosome. And so this is a human cell from a human cell line. And these spots you see are associated with the enzymes that make purines, the things that go into DNA and RNA. And this cell here has been starved for purines. And when it's starved, all of the enzymes of purine synthesis form together into these phase-separated reaction factories where new purines are made. And then if the cell is fed purines, these fall apart, they disperse. And this raises kind of another important aspect of these phase-separated bodies, which is unlike the membrane-bound organelles, that the nucleus is there and it just stays there. The mitochondria, by and large, are there and stay there. These phase-separated bodies are very dynamic. They come and go as needed by the cell. And in general, what's happening is that the cell is chemically modifying some of the components here very quickly that can cause them either to have the interactions that bring them together, or based on chemical modifications like phosphorylation, which changes the charge, means that they're no longer physically held together and they fall apart. And so it raises a whole new level of the biology in the sense that not only are there many of these, but they're also very dynamic. They're very much changing in time in response to different inputs and different needs of the cell. So this is what's happening in vivo. One of the challenges here is that these are very complicated systems. So, in the pyrenosome, there are some 6 or 7 different protein enzymes that are all coming together. In some of these condensates, say in the nucleus for transcription and elsewhere, there are hundreds of different proteins and RNAs that are coming together to form these droplets. Now, frequently, it's not required to have all 100 there. There are a few that are dominant, a few proteins or RNAs that by themselves are able to engineer phase separation. And so one of the productive directions that has been pursued recently is to extract those few components from the cell and study the phase separation specifically of those components. And so I want to transition now to talking about some in vitro experiments that were done in the lab of Mike Rosen. And this was really inspirational for me and my group getting involved in this, because as theorists and modelers, we're a little bit afraid of taking on modeling something that has 100 different components, but maybe one component or two component we can get our heads around and start trying to figure out from a physical perspective how to understand this phase separation. And so
so Ned, just very quickly,
yeah.
Would you consider these examples as equilibrium or more like non-equilibrium phase transitions? I mean, ultimately it's biology and it's probably to some degree non-equilibrium, but do you try to make a distinction here or not really at
Yeah,
this moment?
absolutely. I would make a distinction. And I would say that essentially these structures are equilibrium structures. The dynamics is driven by energetic processes in the sense that, in order to make the pyrenosome, energy is used to phosphorylate certain components. But once they have those extra phosphate groups, then it's just an equilibrium process by which they diffuse and bind to each other. That's in contrast to systems that people really think of as more active matter, where I have cells or motors, things that are actively moving around, and the phase separation, as soon as the energy source disappears, immediately the system will fall apart. This is more like cells that are aggregating this way, or motors that are forming some kind of complex structures by bringing polymers together. So I think my take here is that, if I can say, turn off the energy source, and yet these things will at least transiently hold together until sort of the phosphate groups fall off, then I should think of this as at equilibrium. Now, that said, there's a kind of crossover where you look at, well, what about the sizes of these objects? Here I see something where there's a lot of small droplets inside a big droplet, but maybe the true equilibrium of this is that these droplets would merge, right? Because they have more surface tension when there are a lot of small droplets than one big one. And yet, you haven't waited around long enough for the system to reach its 2 ground states, so you're still in some kind of metastable configuration. And that actually could be relevant. Again, the same kind of thing is happening here in the pyrenosome. You see lots of small droplets. Maybe the true equilibrium ground state of the system is one big one, but the system hasn't had time for these droplets to ripen and merge. And so you are in that sense in a sort of crossover. It's equilibrium physics, but you haven't reached the true equilibrium ground state.
So maybe I would like to make a comment. So you mentioned phosphorylation. So phosphorylation could be an ATP-driven process, and then the dephosphorylation may or may not be ATP-driven. So that by itself, if you allow this kind of ATP-driven loop, could potentially make the sort of the modification dependence phase separation non-equilibrium, right?
Yeah. So yeah, I mean, now you're again getting into the idea that there's not such a clean distinction between these two, because you could imagine that I have phosphorylation happening outside the droplet. So components become sticky, they go into the droplet, but there's no phosphorylation happening there, at which point the phosphates fall off or there's a phosphatase. And so there's a cycle that actually could maintain the size of droplets. This is a very active field. The question of what sets the size and distribution of these phase-separated droplets inside of the cell. So again, you get to a regime where, yes, it's not exactly equilibrium, it's not exactly non-equilibrium, it's some kind of crossover. These are great points. Okay, so let me now continue on to these in vitro experiments from Mike Rosen and his group. And as I said, these got us interested because we felt that they were perhaps more tractable for theory and modeling. And so here we go. So this is a system that's engineered based on 2 domains. And if you look here, you'll see what these proteins look like. Of course, this is a schematic, but these green circles represent something called a sumo, which is actually a small folded protein domain. And you have these in your body on various proteins. And what they do as part of their function is they interact with a small peptide motif, part of another protein sequence. And the sumo domain has a binding site, and it binds onto this sim, which is this sumo interacting motif. And as I said, this is happening right now inside your cells. Sumos are binding to sims and moving things around. But Mike Rosen took advantage of this to engineer proteins where there was a string of sumos on these polysumos and a string of sims on these polysims. And these were able then to phase separate. And the idea was that they wouldn't phase separate on their own. The sumos don't interact in particular with the other sumos, and the sims not with the sims. But once you put them together, then you would find phase separation. And so these were just some images showing, different concentrations of the polysumos and different concentrations of the polysims. you would get phase separation, and they were colored in different ways. And if you looked at them together, you could see, okay, even though they're all phase separating, here if I have more polysumo, the droplets are enriched for the sumos, and if I have more polysims, they're enriched for sims. But you could go beyond this region where everything phase separates and look at the phase diagram. And so at lower concentrations then of the polysumos and polysims, the system would not phase separate. You would see no droplets there. But as you increased both of them together, eventually you get to a phase boundary. And everywhere in this region is a two-phase region. So you'd see what you saw on the last slide, that there were droplets that contained both the polysumos and the polysims. And clearly, if you just go along in this direction and you have more and more of the sumo or more and more of the sim, it would not phase separate. And so the picture that arises is that you have, these interactions that are multivalent. So one polysumo could interact with many polysims, one polysim with many polysumos, and you would form this network that would actually hold these molecules together, causing them to phase separate away from the surrounding medium. So we became interested in that and wanted to try to understand a little bit better what was driving this phase separation, what was giving rise to this specific phase diagram, and also things like the concentrations of the two in the different phases. And so in order to do that, we had to learn some polymer physics. And so I want to share with you what we had to learn at the very beginning of this. So I want to take you back to school for a moment to Polymer Phase Separation 101. And so this is the standard picture of polymer phase separation, at least the simplest theory for this, which is due to Flory and Huggins. And the idea is extremely simple, that you have some free energy density of this polymer solution. And for the moment, we're just going to write down the free energy density for a homogeneous system, not yet thinking about it phase separating, just saying if it was forced to be homogeneous, what would the free energy density be? And you start with these two terms here, which have to do simply with the entropy of mixing. This is the number density of the solvent, say water, the volume fraction of the solvent, the number density of polymers, how many polymers there are per unit volume, and the volume fraction of the polymers. And these terms by themselves would give rise to a free energy density that had this upwardly curving form as a function of the volume fraction of the polymer. And what this means is that basically nothing interesting happens in this system. You give the system a certain density of polymers, the entropy of mixing says, hey, we get more entropy if these things are just spread out through the entire solvent. That minimizes the free energy, and the system just sits there being homogeneous. But then there's this extra term here, which says that polymers and water, polymers and solvent, interact with each other in different ways. So it might be that the polymers are maybe hydrophobic. They have oily groups attached to them. Think about proteins that have side chains that look like little bits of oil. And therefore, there'd be a term here that measures the interactions combined polymer-polymer, solvent-solvent, and solvent-polymer into this one parameter chi. And if this parameter is positive and large enough, then the free energy as a function of this polymer volume fraction gets this different shape. This is simply adding this term to the expression we had before. And now you see that there's these two minima of the free energy density. And what this means is that everywhere between these 2 points, here's the free energy of the homogeneous system, but this line actually gives the free energy density of an alternative system in which part of the material has phase separated into this polymer dense phase, and the rest is a dilute phase that is mostly solvent and few polymers. And so what that means is that everywhere between these 2 points, if the polymer volume fraction lies between this, the true ground state of the system is phase separated. So it would form, for example, in this regime, droplets of this dense phase surrounded by the solvent. Of course, we're not paying attention here to the surface tension at this point. In fact, it wouldn't be many droplets. The true ground state would be one large droplet of the polymer-rich phase embedded in the dilute phase. So this is the very simple idea that you have a competition between this entropy term, which is favoring everything being homogeneous and well mixed, and this energy term, which is favoring separating the polymers from the solvent. Now that's really all we need, but I would be remiss, having gotten this far, not to show you one more thing about polymer phase separation, which is the question of whether this will happen spontaneously or not. And so what I want to point out is that everywhere in this region, around here, the local free energy density is curved downward. And that means that the system is actually unstable to even locally forming slightly more dense and slightly less dense regions of polymers. So if you start the system and quench it right here, everywhere throughout the system, it will start to phase separate, and eventually you'll end up with droplets of this density and droplets of this density. But there's another region here where the local curvature is upward, even though it's actually in the two-phase region. So globally, it would like to phase separate, but locally, it's actually better to have it be homogeneous than locally break up into a dense phase and a dilute phase. And so this region called the spinodal region means that you actually need to nucleate the dense phase somehow. It's not going to happen spontaneously. Somehow there has to be some kind of fluctuation where a bunch of polymers just by accident get together to create a local region of very high density. And only then sort of does the system realize that there's an alternative ground state where the polymers are phase separated. And so that gives rise to this following picture where everywhere between these 2 points, where the inflection point, the system will spontaneously phase separate. But out here, phase separation would have to go by nucleation. And this is, of course, a very important thing in biology, where the cell can control where phase separation happens based on nucleation. And of course, cells have evolved all of these mechanisms that don't leave this to chance, where there are nucleators inside the cell or local regions where phase separation is preferred to happen. That's the basis for an entirely different talk. I just want to leave you here with the thought that the overall phase diagram then looks like the following. As a function of the polymer volume fraction here, if the interactions are weak, then nothing interesting happens. It's just one phase, homogeneous. But if the interactions are strong enough, you enter this two-phase region. So if you start adding polymers to the solvent, initially those polymers, there's not a high enough density of them to phase separate, entropy wins, and it's just a homogeneous system. But as you keep increasing the density of polymers, you finally come to this phase boundary where at least the true ground state says that any additional polymers that you put into the system will tend to form these droplets. The surrounding material, though, will be stuck at this concentration of polymers. So it's only the additional stuff that goes in that forms droplets at this density. So if you keep moving through this direction, all that happens in this intermediate region is that the fraction of the system that's the dense phase grows and the fraction of the system that's the dilute phase shrinks until finally you reach this point where the whole system is the dense phase. And from then on, it just gets denser and denser with polymers. All right. So that's, the basic picture of polymers that phase separate because of energetic interactions. These are basically polymers that like to stick to each other and would rather be near each other than next to the water. But this has not captured yet the idea of that there are domains that bind to each other in this one-to-one fashion, these sumos binding to these sims. So we wanted to think about how could that compare to this traditional picture of phase separation. And so in order to do that, we did some simulations, and this is really kind of embarrassingly simple, but this is really what we did. We said, well, this phase separation property, it's not specifically about 3 dimensions, and 2 dimensions are easier to visualize. So let's start by doing some simulations of our Sims and Sumos on a 2-dimensional system. And again, to keep things simple, let's put everything on a lattice. We don't have to worry about all the possible conformations then. And so this is exactly what our system looked like, where the little blue squiggles are the polysumos and the little red squiggles are the polysims, and they all live in this 2-dimensional lattice. And the only rule at the moment is that the sumos can't overlap with themselves and the sims can't overlap with themselves. And it doesn't look like much is happening here, but that's okay because there's no interactions turned on in this case. This is like 2 layers one of polysumos, one of polysims that don't know about each other. But then what we can do is we can turn on the interactions and say, well, what if we now turn on a strong interaction, meaning that when one site is occupied by a polysumo monomer and one by a polysim monomer, that's favorable. So they like to start overlapping with each other. And then what happens is the following. So clearly something has happened, but it's not entirely clear by looking at this what has happened. So we stare at it for a little bit, and you start to realize that there's kind of two different zones in this simulation snapshot. There's a region like this here, which is pretty dense, and they're both polysims and polysumos, but they're kind of doing different things. Each one is kind of interacting one polysim with many polysumos and vice versa. But then there's this other kind of thing that can happen where there's a more dilute region where the polysims and the polysumos get together in a one-to-one way. And so they interact like this, and they really form a partnership where they just overlap perfectly with each other. And so you start to realize that, well, this is sort of looks a little bit like phase separation, but it's not clear that it's exactly, phase separated. But then we say, well, look, this is the only interaction. What if we had a tiny additional little bit of interaction that would favor the polymers being near each other? And so if you do that, adding just sort of, a fraction of a KT per neighbor, then you get something that looks a lot more like a phase separated droplet. And it's pretty much as close as we're going to get to phase separation in this very simple 2D square lattice model. But I want to emphasize first something, and we can actually go back for a moment to this picture, because we got used to this in this little lecture about phase separation, that it was energy and entropy that were competing. But if you actually look at these two different configurations here, you start to realize that really, whether it's this denser phase or this more dilute phase, that all of the bonds are formed. That is, every single polysumo site and every single polysim site have somehow found each other. So the energy is actually quenched. The energy in this system is a minimum, both in this dense phase and in this dilute phase. So you say, well, hold on a second. If the energy is the same, then what's driving the system to phase separate? And the answer is the only thing that's left, which is the entropy. And so if you look at this, in this system, the polymers are dense, so they don't have a lot of free volume per polymer. Their translational entropy is very low, but their conformational entropy is high. That is, that each one of these polymers can do its own thing. The polysumos can do one thing, and the polysims can do something else, so they have all of their conformational entropy. By contrast, in this dilute phase, this has a higher translational entropy. It has more room to move around, but you'll notice that once you've determined what the blue polymer is doing, the red polymer is constrained to do essentially exactly the same thing. So one of them has given up all of its conformational entropy. And so in this phase, you have a lot of translational entropy, but very little conformational entropy. And the phase separation then is driven by this competition now between these two types of entropy, rather than a competition between energy and entropy. So in this sense, it's a somewhat different type of phase transition. Now, I showed you that you can get this to phase separate by a little bit of non-specific interaction between the polymers, but you should always be skeptical and say, maybe I'm cheating you, maybe that was all that was required to hold them together, and it's all about energy. But you can test that by then turning off the specific interaction and asking what happens. And indeed, the system falls apart. So it was really critical that these polymers had these strong one-to-one interactions in order to get the phase separation. So it really was driven by this strong multivalent one-to-one interactions that characterize this Sim Sumo system. So there's much more to say about this, but I want to spend some time talking about the Pyrenoid, because I've come to love the Pyrenoid. So let me, at this point, conclude on the Sim Sumo modeling and review the important things that we've learned, which will also be important for thinking about what's going on in the Pyrenoid. One is that phase separation can be driven entirely by entropy, in this case, by translational versus configurational or conformational entropy. And second, that the low density phase was dominated by dimers. We noticed we never saw a lone polysumo and a lone polysim, even in the dilute phase, they were still interacting. So when you think about where the phase separation is going to happen, at what concentration, and so on, we really have to think not just about the dense phase, where these polymers are interacting in a kind of promiscuous way, but also what's happening in the dilute phase. And that's going to be an important point for thinking about the Pyrenoid. So this is a moment when I could pause. If there are any questions, I'd be happy to answer those.
Maybe a quick question about the scenario where you say they phase separate based on just the different type of entropies. So if you go back to that slide, I don't see a well-defined interface. So one more back. One more back. Not this. Yeah, this one.
Yes.
So I don't see a well-defined interface. I don't see a circular, let's say, a circular drop. But when you do the simulation, you know it's phase separation because you can see nucleation, and then eventually everything coalesce or just coalescing into one big drop.
So this is a great point. So what I haven't shown you is sort of the thermodynamics here. So we can study things that reflect on phase separation in terms of how the free energy of the system behaves as a function of density, and so on. But you make a really great point, which is that this system doesn't really round up, and what really causes the systems to round up is surface tension, right? That there's an energy of this interface between the dense phase and the dilute phase, and that the higher that is, the more the system will be confined to spherical droplets. And so, as I said, in the usual case, you have this energy of interaction. You have sort of, and at the interface, the polymers are exposed to the water, and that's very energetically unfavorable. And so characteristically in oil and water, you have this high surface tension. Here, because the energy is all quenched, what's giving rise to surface tension is rather subtle. It's a kind of limitation on the conformational entropy at the interface between the dense phase and the dilute phase. It's a much weaker source of surface tension than an energetic source. And that's why you don't get these very well-defined droplets. It's because of the low surface tension. And I have to say, this is a super interesting topic in the context of these biological condensates, because typically they're measured to have quite low surface tensions. Something like 1 kT of energy over a region that's 10 nanometers by 10 nanometers, which is crazily low compared to oil and water. So part of the interesting question is in the biology is, to what extent are the interactions tuned to give rise to these sort of weak surface tensions? And really people don't understand whether or that's important, whether that's an epiphenomena. It's a very active area of research. So that's a great question. Thank you for bringing that up.
Maybe then I would like to follow that up. I mean, if it looks like this and it doesn't coarsen because there's zero surface tension, let's put it to the extreme limit, then it would be a homogeneous phase with potentially higher fluctuation of density. Then it wouldn't be phase separation if we take that point of view.
Yeah, absolutely. If the surface tension went to, I mean, that's a characteristic of the critical point for even the phase separate, is that the surface tension goes to zero at that critical point. So there is a surface tension here, okay. And we can argue for that in other types of simulations, but it's extremely small, and that's exactly why it, by eye in this small system, you're like, I'm not sure that's phase separated, but take it from me, we have other tests of this, and of course, the biological systems, by and large, operate farther from the critical point. There is some additional interactions that tend to hold them together, even though, as I've said, the surface tension tends to be weaker than we're used to in sort of more traditional physical chemical systems.
Okay, thank you.
Awesome. All right, so I'm going to push ahead then in vivo. We're going to dive inside the cell, and specifically, we're going to dive inside of the cell of algae. And so these are guys, in this case, living out in the ocean. They do all sorts of interesting things. They swim around, they do photosynthesis, but I want to focus on this particular aspect of this particular algae called Chlamydomonas, which is, as I've said, one of these many systems that is fixing CO2 out of the atmosphere. And so this takes place in this organelle called the pyrenoid, which is highlighted here in these different organisms. And again, this is one of these non-membrane bound organelles. And my group got interested in this because of a professor at Princeton, Martin Jonikis, a young fellow who is extremely interested in the pyrenoid from the point of view of improving crop yields, because it turns out that this system is extremely efficient at fixing CO2 because it concentrates CO2 inside the pyrenoid. And this trick that's used by algae and some plants is not universal. So a lot of our food crops like wheat and corn don't take advantage of these carbon concentration mechanisms. And Martin's goal is basically to teach wheat and corn to have pyrenoids. Now, that's a long-range goal, but along the way, there are a lot of fascinating biophysical questions. And so we've been having a great time working with Martin, trying to understand some of these aspects. And I'm going to tell you now today a little bit about what's going on that's holding the pyrenoid together. And so here's a blow-up of our hero today, Chlamydomonas reinhardtii, or clammy. And this is just an electron micrograph, false colored here, so you can see the chloroplast. This is where the photosynthesis is happening. This system is using the energy from sunlight in order to fix CO2 out of the atmosphere. And that's happening inside this pyrenoid here. And so what's in the pyrenoid is largely this enzyme ribosome. And this had been known for many, many years. There's this large enzyme complex. I've shown you a picture here. It has sort of 8 large subunits, 8 small subunits. And ribosome is ubiquitous. It's basically what's fixing CO2 in all plants and algae. But it's held together in the pyrenoid not by a membrane. There is no membrane. There are some starch platelets around it, but there's holes in these. And so kind of question that had not really been addressed is, well, what's holding the ribosome in there? If it's free to diffuse out and there's no membrane, then why doesn't it leave? And so that was a question that was addressed by Martin, and he was developing genetics and genomic technologies for dealing with this organism. And by virtue of those, he was able to discover that there's another major component. It's not just ribosome in the pyrenoid. It's also this protein, which he modestly called EPIC1. The P is for pyrenoid, which is an intrinsically disordered protein. That is, left by itself, it does not fold up like ribosome or most of the proteins we're used to into a particular form. It's basically a floppy polymer. And actually, this is kind of ubiquitous in these phase-separated bodies, that many of the proteins there are either disordered or have a disordered regime that play an important role in holding together these condensates. And in this case, the EPIC1 was then discovered to have 4 nearly identical repeats. The sequences are very similar in these regions. And it was very clear that if you remove EPIC1, then there is no pyrenoid. It falls apart. And it was also possible, I haven't shown that, to reconstitute this system in vitro, that riboscomes don't phase separate, EPIC1s don't phase separate, but you put them together and they phase separate. And that's definitely happening inside the cell by fluorescently tagging the EPIC1 and the ribosome. You see that they co-localize inside of the pyrenoid, and they're actually holding each other together. So it's very much like the Sim Sumo system, where one component is this ribosome, and the other component is this EPIC. And in this case, one of them is a floppy polymer, the other one is a more rigid object, which has its own implications. But I wanted to focus on some other aspect of this.
Ned,
yeah.
Can I just ask very quickly, what is the purple color here? Is
the purple
in the
color,
system, do you still have these starch components around it, or what is labeling here in purple?
Yeah, I think this is just a label of the chloroplast. Yeah, this is chlorophyll.
Right.
So you're seeing the fluorescence from the chlorophyll. So this algae, it's doing photosynthesis, so it has a chloroplast. It's one big chloroplast. It's sort of a significant volume of the whole cell is this chloroplast, where photosynthesis happening, and that energy is being used, at least in large part, to fix CO2. Because these guys, they don't get to go to lunch and have a sandwich or something like that. If they want to get if they want to grow, they need the carbon from CO2. So a lot of their energy is going in to taking that inorganic carbon in CO2 and eventually turning it into sugars and then into amino acids, into nucleotides, all the building blocks to make a new algae. Yeah, good point. All right, so yes. So what can we say about this other than this is one of these two component phase separated systems? That's nice. Great to see one of them is actually playing an important role out in nature. One of the things that is characteristic of these systems is that they are liquid, and that was also demonstrated then for the Pyrenoid. In this case, by something called fluorescence recovery after photobleaching. So by virtue of being able to make variants of EPIC1 or rubisco that are fluorescently tagged, you can then bleach the fluorescence with a high intensity exposure, and then ask whether this then recovers or stays with one of these Pyrenoids half bleached. I should say for some time it was believed that the inside of the Pyrenoid, the rubisco, was in a solid state, but these experiments showed that it was liquid. Because over the time scale of about 2 minutes here, the bleached versions of rubisco and the unbleached ones all mix up. So the system is liquid. And this actually has biological implications because there are some proteins inside of the Pyrenoid whose job it is to rescue rubiscos that have basically gotten stuck. That instead of trying to fix CO2, they've gotten stuck with an oxygen in their active site. And by virtue of being liquid, these enzymes, these sort of repairmen, are able to diffuse around and find different rubiscos to fix. From our point of view, though, we're interested in another aspect of this Pyrenoid, which has to do with what happens during cell division. So let me just quickly then say, here's the movie of the bleach and the recovery. So this is a liquid droplet. But the interesting thing that Martin found by watching these is that the Pyrenoid, even though it's one of these phase-separated droplets, has its own dynamics, not just its internal mixing. And so what Martin noticed is that prior to cell division, so this is an algae, like any living organism, it has to reproduce, it grows and eventually divides, and that division is there's a cytokinetic ring that pinches all the way through the cell, through the chloroplast, and eventually pinches through the Pyrenoid. But actually, prior to that pinching, something interesting happens to the Pyrenoid, and I'll show you that. That here is just a few minutes before this cell division is going to happen. A great deal of the EPIC1 and rubisco have left the Pyrenoid and spread out through the chloroplast. And this is actually transient, so that as soon as the cell divides, now it's divided into two, and this is rotated because the cell's alive. It actually just spun around here. Those then recondense into the two daughter Pyrenoids, and this happens reliably. So it goes through another round of division, there's a spreading, and then a recondensing of these molecules, which is a little odd because, of course, if this is held together by phase separation, by physics, physics hasn't changed in these 7 minutes, right? Physics has not varied. So what is it that's causing this system that was sort of strongly phase separated now to become, enter sort of at least partly enter this one phase region or go closer to the phase boundary so that more of the material is dispersed? So, of course, there are both not only how questions, but why questions. Usually in biology, we tend to punt a little bit on the why questions. Well, does something for the cell, evolution. In this case, it's actually probably easier to answer the why question, because one of the things that's noted is that occasionally when this cytokinetic ring tries to pinch through the Pyrenoid, instead of it dividing in half so that each daughter gets a Pyrenoid, sometimes this goes wrong and the Pyrenoid gets squirted out to one side. So one daughter gets the Pyrenoid and the other one doesn't. But as a result of this dispersal of material, even the unlucky daughter has a bunch of EPIC1 and a bunch of rubisco that then can condense again de novo, forming a new Pyrenoid. And so even that unlucky daughter can start doing photosynthesis of carbon fixation. So that may be the why, but the how is then still pretty mysterious. And this stuff happens very fast, right? This is happening. Well, this movie is sped up, but over a period of a few minutes, much too fast for this to be sort of new protein that's going in. It's really the same protein molecules that are happily condensed, decide to disperse, and then come back together again. And there's the movie showing, this process. You can really see the spreading and coming back together. So what can we say about that from the point of view of theory? Well, we don't know yet exactly what's going on, but there's something interesting about these multivalent systems, which I want to tell you about in the last, couple of minutes here. And that has to do with the relative valence of the two components. So in the Polysumo, Polysim, we looked at things that had the same relative valence, 10 and 10. Here we have these epics that had these 4 repeats, and the ribisco has 8 repeated subunits. And so it's sort of, well, does it matter that 4 is somehow related to 8? Well, let's explore that a little bit. So let's start by, as theorists, we can do anything we want. Let's consider what it would be like if epics were length 3 or length 5. And what we're going to do is the same kind of really embarrassingly simple lattice simulations where now instead of polysumos and polysims, we have epics and ribiscos. An epic is just a little polymer occupying 3 or 5 sites on the square lattice, and a ribisco, since it's really rigid, and it has 8 subunits, it's going to be a little block, and it's going to be 4 by 2 in order to get 8 sites. And so you see what happens is if you mix together epics of length 3 and ribiscos at high enough concentration, they do the same kind of thing. This is basically as good as phase separation as we can get. And you can see that if you do the same thing with the same number of monomers, but string them together into length 5, you also get phase separation. And here you look at something which is more or less like a phase diagram based on the sizes of clusters. This is as you get higher and higher density of material. This is as you get stronger and stronger interactions. You can see that there's a phase boundary. As I increase the amount of material, I get big clusters. And as I go from L equals 3 to L equals 5, the phase boundary moves to the left. That is, and that's not surprising, L equals 5, basically there's more sticky sites per polymer. Each polymer still only has 1 center of mass degree of freedom. So more sticky sites gives rise to a greater tendency to phase separate. And you see the phase boundary is now moved to lower concentrations. I need less material to phase separate if the epics are length 5. So what about length 4, which is what, the actual number of repeats? Does it just interpolate between the two somewhere in the middle? Well, that would be boring. So in fact, what happens when you have epics of length 4 is that the phase boundary gets shifted way up to high concentration. This system basically has to be filled with epic and ribisco before it'll phase separate. And if you look at the snapshot, you can immediately see what's happening. And what's happening is that because 4 plus 4 equals 8, there's an alternative configuration of epics and ribiscos, which is energetically satisfied. That is, I can mix 2 epic 1s with 1 ribisco, and all of the binding sites on the ribisco are occupied by epic 1s, and all the binding sites on the epic 1s are occupied by ribiscos. They formed a little happy family. And these little happy families don't need to interact with each other, right? They're perfectly satisfied. In a way, this is kind of familiar in physics as a kind of magic number effect that we're certainly used to the periodic table where you look off to the right and you see, oh, these are the noble gases. They have just the right number of electrons to fill all the shells, and they don't need to do chemistry, right? They're the noble gases because the nobility didn't need to mix with anyone else. Well, these guys are like the protein version of the noble gases. They don't need to mix with anyone else because they've sort of filled all of their orbitals. They filled all of their binding sites. So it's a kind of biological magic number effect. And so, one of the speculations then is that, this might have something to do with this very rapid transition between a phase separated domain and a non-phase separated domain or regime that I'll touch on. But before I conclude with that, let me say again, you should always be skeptical. So I've done something here which looks okay, but it's in 2 dimensions on a square lattice. Does this argument about magic numbers generalize?
Hey, Ned, before you move on, can I ask you something quickly?
Yeah, absolutely.
Yeah, so in the L=5 diagram, in the phase diagram at the bottom right,
yeah,
you seem to form clusters at high concentrations, clearly, but also at zero specific bond energy. Is this because also you have this non-specific binding, weak non-specific binding, or is it some other sort of jamming going on?
So this is actually really getting up to high concentrations. So what's happened here is, okay, this is admittedly a sort of naive view of clustering, and again, I'm not showing you the thermodynamics. This just says if one of these sites of the ribosome and an epoch 1 touch each other, they're part of a cluster. And so even if the energy goes to 0, if I throw a bunch of guys down on top of each other at high enough density, just percolation will give rise to big clusters. So this is effectively saying, okay, you got to be, this is really just a percolation cluster that's formed here. And as I said, we do better than that, but this was a simple way of showing what's going on. And down here, of course, this is phase separation driven by these interactions, again, with a small amount of non-specific interaction. But up here, there's 0 interaction. They're just percolating.
Yeah, maybe another quick question. So when you go to the bottom with high specific bond energy, it looks like the transition is relatively gradual. And I suppose, does it have to do with the fact that you look at a small system, and what would happen if you now go to basically an infinite system, a large system, and would you see then a first order type phase transition? What would you expect that?
Exactly. You're absolutely right. So for these small systems, it's entropically favorable in the small systems to have, a few clusters, whereas if you go up to a macroscopic system, there would be one macroscopic cluster. And so there would be an absolute, mathematically precise boundary where you went from having no macroscopic cluster, to a first order to a transition to a system that had, one macroscopic cluster. So yes, so this gradualness is not a signature of second order phase transitions or large fluctuations or something. It's just, as you said, a consequence of doing this with small finite systems. So, but as I said, we've done more than this. This is particularly nice because it's visually very clear what's going on. But again, it's important to test the generality of this. There's nothing about what we've said that is specific to 2D or to the fact that this is on a lattice, but nevertheless, we've generalized this to think about something which is more realistic, where it's in 3D, it's off lattice, and here, getting away from the notion of rigidity because of the focus here was just on polymers. We have something that looks more like this Polysumo Polysim system, where I have these 2 polymers, and now you can see again that okay, if you put these together and let them equilibrate, you can again get a phase separation here where there's a dense phase of we'll call them polysims and polysumos, each one interacting with many partners, and a dilute phase where they've formed these heterodimers of one polysumo binding to one polysim. Down here is just a study of what happens when we start to vary the stoichiometry. What if we have more polysumos than more polysims? And you can see lots of things happen. The dense phase spreads out because there's more unbonded monomers there. This is just to give you a flavor that there are lots of things that you could then study that would actually have implications for the biological system. And I'd be happy to chat with people more about that afterwards. But the basic notion here, though, is that these magic numbers definitely control the phase diagrams. That is, it's a big difference when the valence of these is equal or equal within a rational number like 4 + 4 = 8, than if they're actually, not, related to each other in that simple fractional way. And so, for example, here is a plot focusing on the black curve, which is a low temperature. of what happens if I have polymers of length 14 and I mix in other polymers of different lengths. And what you can see here is that if I actually mix 14 and 14, I have to go up to relatively high concentration to get this system to phase separate. But by contrast, if I mix in, 14 and 11, this system phase separates. I'm having trouble snapping, but in a snap, like, this system, really loves to phase separate. And it's because in this case, the condensate is competing with a very, very stable state of a dimer that's fully satisfied. All 14 sites on this one bind to all 14 sites on the other. Whereas with 11, there's no good combination of 14 and 11 that satisfies all the bonds. So this system is strongly driven to phase separate in order to minimize its energy. So really, I've come to the end of what I had planned to say today. So the last question is still speculative, and this is something we're actively working on with Martin, is to what extent do these magic numbers have anything to do with the Pyrenoid? We still don't know. Real biology is always, more complicated than our simple models, but it's giving us some directions to perform experiments on. And the idea would be that in the condensed phase, there's no such, magic number agreement. Maybe the epochs are length 3 in effect, or maybe there are more binding sites that we know about, but there's no happy small system that can separate out like a 4 + 4 = 8. And so the system is driven to phase separate. But then when it comes time to divide, by simply chemically modifying, say, one site per epoch, a very small, low-cost chemical modification, you could enter into one of these magic number conditions, at which point all of these trimers of 2 epoch 1s and 1 ribisco have no longer any need to interact with the others, and so they can spread out. They can become the noble gases, spreading out like a gas throughout the chloroplast. So this is the idea that we're exploring. So by way of conclusion, I hope I've at least stirred your interest in these non-membrane bound organelles, biomolecular condensates that are ubiquitous inside our cells. We have lots of these inside our cells right now, dynamically forming and breaking up. That in the case of these multi-component multivalent systems, the phase separation can actually be driven or primarily driven by entropy, which is not so familiar in non-biological systems. My favorite take home, though, is that the small oligomers are very important. We should not ignore those small oligomers, dimers and trimers and so on in the dilute phase, because they are really the thing that's competing with the dense phase. And we've had fun exploring this magic number effect, which might have something to do with the Pyrenoid, but we'd love to see also people trying to produce magic number systems, whether they're in vivo or in vitro. I would certainly love to chat with people about that. And so finally, I want to acknowledge the people who did the work, my student Bin Zhu and postdoc Yaojun Zhang, who really did all of the heavy lifting, but we've had lots of great collaborators both at Princeton and elsewhere and funding. And then I want to just thank you for giving me the opportunity to chat about this. And I look forward to answering any questions.
Thank you so much. Thank you, Ned, for this fantastic talk, very inspiring, really fantastic work. Yeah, so if there are any questions, please, it would be a good time to ask. While maybe people think about questions, I was wondering now about, you mentioned how this could be used in the ribisco system at the very end. During cell division, this thing has to split up, or it has to maybe potentially form dimers to allow the splitting up of these clusters during the cytokinetic ring. So what kind of ideas are around? I mean, does it have to do with phosphorylation potentially to change the valency?
Yes.
I mean, the length of the polymers is not going to change, presumably.
That's
so it has
right.
to be this valency.
Absolutely. So yeah. So our thought on this is exactly what you said. That insofar as the same molecules leave and then come back, it can't be based on cutting the epoch 1s in half. I mean, that would do it, but then they couldn't get back together. There's nothing in the cell that lets you take 2 proteins and join them. And in fact, this phosphorylation is the likely suspect. First off, there's evidence from proteomics that epoch 1 does get phosphorylated. So there's definitely, variant EPIC1 peptides that have phosphate attached, and trying to quantify that is one of the things that's going on now with Martin. And it's also the case that phosphorylation, which adds this charge, has a very, very strong effect on the protein-protein interactions. And in fact, many of the examples of dynamic phase separation in cells are driven by phosphorylation. It's not the only modification that plays a role, but typically it is modifications that change the charge state. Phosphorylation, dephosphorylation, adding a negative charge, methylation, demethylation, changing the charge. So that seems to be one of the themes that from the cell's point of view, it's a relatively cheap modification. There's, it's a standard thing, phosphorylation, dephosphorylation, and it also just has a very strong effect on the protein-protein or protein-RNA interactions. So that is our best guess. And I have to say, honestly, that's independent of whether it's magic numbers. It's almost surely, phosphorylation, maybe some other chemical modification, but there's really nothing else, right? Like it can't be cleaving. The riboscos, they really are not modified, and they have to do their job, as in fixing CO2. So it's very likely this EPIC1 that's getting modified.
Yeah, I mean, there was a question in the chat box actually about EPIC1, if the number of binding sites have been measured somehow. I mean, is it clear that the number of binding sites might be 4 or changing during cell division?
Yeah, so, the jury is still out on this. It looks like there's actually probably an additional binding site in addition to the obvious repeats at the C terminus. This is actually kind of better from my point of view. I didn't want to go there, but then it would have 5, it would sort of normally have 5 binding sites, which would be sort of strongly in the phase separating regime. And then, by removing one of them by phosphorylation, you go to 4. And that's actually good because it pushes you in the right direction. It both makes the polymer shorter, which tends to be bad, make things fall apart. And by being 4, it's 4 plus 4 equals 8, so you get the magic number effect. So we're crossing our fingers that the native state is 5, and then modification brings you down to 4. There is information, and it's very clear that the motifs I showed you do stick to ribosco in a particular location. But there's also probably additional interactions because the energies that are measured for that are still relatively low, and they don't explain some other measurements about, the overall binding affinity of an EPIC1 for a ribosco, which I have to say raises an interesting question about, the linker length. When we do things on a lattice, everything has a linker length of 1, sort of by definition, and it all lines up. But it's an interesting question whether the EPIC1s are long enough to stretch between 2 adjacent sites on the ribosco or not. And so one of the things we're exploring is to what extent nature has tuned this linker length, to fit a magic number condition or not.
Mm-hmm. Yes, thank you very much. I mean, there was another question on the chat box about if similar systems about phase separating systems have been designed ex vivo. So I think you mentioned things. I think you're talking about single cell organisms, right? The algae, and about taking things out in vivo, I suppose. That would be called in vivo. I don't know, ex vivo.
I've certainly, one thing to say is like this Sim Sumo system. Okay, Sumo is a natural protein, but this was totally engineered. This thing of 10 sumos and 10 sims, that doesn't appear in your body, right? That was engineered by Mike Rosen. Typically, these systems that have been studied in the protein world, something is borrowed from nature. They're, 2 domains that are known to interact, biologically, are then used as the basis for some designed polymer. But you can definitely do other things, other systems where the design part is more straightforward. So for example, DNA origami, right? Where you say, you can build something and people do lots of, amazing things, where they make, stars and other things of DNA that have very specific sequences. And then exactly how to make them pair up because it's just Watson-Crick pairing. So one of the things that, we'd love is there's someone who was a DNA origami guru to come in and say, all right, well, we can do this, designing something with 4 sites and something with 3 sites and seeing if that phase separates, but 4 and 4 doesn't, it forms the magic number case. That's not something that a capability either for Jonica's lab or for me, but as theorists, we'd be excited to interact with anyone who wanted to try to design systems like that.
I have a quick question. Well, maybe you can go back 2 slides where you show the between the division and the here. So yeah, great. So in the bottom right plot, it's clear it's not isotropic. So there's a pattern to it. Is it just because of the imaging? Is it because of shell shape? Or
okay, this is the chloroplast. Yeah. So the idea is that the chloroplast has a membrane. It is one of these membrane organelles. So this is this is an organelle within an organelle. So so what you're seeing here is that the rubisco and the epic have spread throughout the chloroplast, but what you can't see here is that the algae is actually bigger and surrounds this whole thing.
Okay.
Yeah.
Okay.
That's going on there. I mean, it's another interesting point actually. This shape here is kind of characteristic. What tends to happen is even, you see, this hasn't gone through cell division yet, but it's actually like it knows it's going to have to be divided in 2, and it's stretching out perpendicular to the direction that it's going to be pinched, right? It's going to be pinched along this axis, and it already knows it's getting ready, right? It's getting ready to be pinched in half, and we really have no idea how that works.
So that's very interesting. I mean, the fact that it is sort of dispersing anisotropically would suggest it's not a thermal process, right? If it's just purely thermal, you would expect a isotropic expansion, dissolution.
Well, so what I think is that like the rubisco and epic out here, that's probably just diffusion, right? But this part, this could be, non-equilibrium. There could be, motors or things pulling on it. I mean, whatever you imagine is possible at this point. But again, there's a lot of biophysics that goes on here because there's these starch sheaths that are surrounding it, and that seems like a stable configuration. So the question is, how do you then disrupt that? Yeah. There are lots of interesting things that happen because Martin, for example, has mutants where instead of 1 Pyrenoid, you get like 10. So it's clearly, there's lots of things that go on in the system that are, have evolved to give you this 1 Pyrenoid, and you start messing around with details, and suddenly, the fact is, you could get multiple Pyrenoids. There's no reason that on a time scale of the cell division that they all end up merging. So there's some magic here.
So you said
and so on. Yeah.
Yeah. So you said you can have mutant with 10 of these organelles. So when during cell division, do they all elongate like this or along a particular direction?
Question. No, I don't think they've looked at that. That's a very interesting question. Yeah.
Okay. Maybe one.
Okay. I don't know the answer. I think that nobody knows the answer to that. I think they haven't looked at that yet. In part because, a lot of the images they have that show 10 are frozen images from electron micrographs. But I think some kind of, I think fluorescent labeling like this, even though they're 10, that you might be able to resolve the sizes. These things are sort of on the micron scale, so it's a little bit, close to resolution, but I think it's doable.
So maybe one final comment. So because your magic number model is purely thermal, it wouldn't, even if you suddenly do a phosphorylation to get rid of 1 binding site, it wouldn't give you an isotropic dissolution of your drug, right? It will have to be isotropic.
Oh, right. Yeah. There's nothing in our model that would give rise to some kind of anisotropy like this. And I really don't think this is like a microscopic anisotropy. I mean, this system really is these rubiscos, which are spheres, and the epic ones, which are very floppy. This is something, on the level of cell biology, where, whatever, microtubules and motors are pulling on this. Also, whether the starch sheath is, itself, by virtue of sort of a surface area to volume effect, is giving rise to this, because it looks like the surrounding of this is still always surrounded by these starch platelets. I guess I'm just highlighting that there are lots of things we don't understand, and there's lots of interesting physics still to explore in these systems.
Yeah. Ned, I have also another comment. Actually, when I saw you talk now, it reminded me of some work Dennis Bray did a long time ago, probably something in the 1990s, sort of computational modeling of chemoreceptor clustering, and you know all about it as well. There was this work where you talked about, of course, the valency of these constituents, protein Ka, Kw, and the receptors being important, and their concentrations to form large clusters. So there was observations, and I think when you overexpress Kw, the glue protein, effectively for the cluster, you get larger and larger clusters. And I think that was also in line with Fred experiments in terms of cooperativity. And if you then overexpress Ka, which is more the bridge molecule, then things fall apart and you form smaller clusters effectively. And that sort of reminded me of your work where you say, valence is important, concentrations are important, and then you get it right effectively to get something sort of meaningful out. Yeah. I just wanted to mention that it's interesting.
This is great, and it's let me make a point here. Oops, which is a great point. So what would happen in this system if you expressed more and more EPIC1, right? You say, well, okay, maybe initially not too much. Maybe the Pyrenoid gets to be a little bit more enriched for EPIC1, but eventually, if there's enough EPIC1, instead of the EPIC1 bridging interactions between ribiscos, each ribisco will be surrounded by its own little cloud of EPIC1s, right? The EPIC1s will bind to that ribisco, and then and now there's no reason for those to interact anymore, right? I could have each ribisco is like a little hairball with EPIC1s coming out of it, and these hairballs no longer want to interact. And in fact, that's exactly what happens in these simulations, that if you go to too high a different stoichiometry, then you lose the phase separation exactly in this way, that you get these small oligomers that form, but they're basically hairballs of one species around the other, and they no longer interact.
I mean, it's the same effect you also have in immunoprecipitation, where you have antibodies and antigens falling out effectively, and you also have to tune the concentrations. Otherwise, you get these adverse effects where you don't get a big condensation of this stuff. So this effect seems quite common. Yeah,
that's great, and I think other people like Mike Rosen has some systems where he's seen things, phase separating great, they go to a membrane, but if you start to get away from the right stoichiometry, it basically kills it for exactly this reason, that you start to sort of saturate one species, the minority species, with the majority, and then there's no reason for it to hold together anymore. So again, it's something that's an interesting question here is what maintains the stoichiometry? Like, is that, just sort of gene expression is tuned to the right place, or are there some kind of feedback mechanisms that, if you're a lone EPIC1 or something, maybe you're very vulnerable to proteolysis. And that's our current hypothesis, is that you're being protected by being partnered. And if you're not partnered, you're susceptible to being degraded, and that would keep you close to the right stoichiometry. Again, another definite direction of interest here.
Very good. Thank you so much, Ned, for your time and for your talk, and for everyone for attending and asking questions. So thank you again, Ned. It was fantastic to see you as well. I haven't seen you for a while.
Likewise, and looking forward to seeing you again in person one of these days.
Thank you very much, Ned, again, for doing it.
It was a pleasure. Take care, everybody.